Dr. Jack Szostak met Dr. Elizabeth Blackburn by chance, but their collaboration paved the way for the discovery of telomerase.
I was at a Gordon Research Conference on nucleic acids in June of 1980, and I had presented on my research on the tetrahymena telomeric DNA sequence and structure. And after my talk, at some point after my talk during the conference, I was walking across a lawn and somebody came up who I'd never met before, and his name was Jack Szostak, and we got talking.
She'd been working on telomeres in this, somewhat unusual organism, tetrahymena. Very different cell biology from the sort of organisms people were more used to thinking about, like yeast and us. And so there was a question as to whether the interesting things that she was seeing were more broadly applicable.
And we were walking across this big lawn at the school, which was the site where the conference was being held. And pretty much by the time we'd got, I think, to the end of the lawn, we'd sort of decided on this exciting experiment, which would be to put the tetrahymena chromosome telomeres onto a linear molecule in yeast and ask whether those tetrahymena telomeres would allow the linear molecule in yeast to survive. Because normally without such, if you just have a linear molecule of DNA in yeast, the experimental findings have been that it would just get eaten up by the yeast cell and wouldn't survive.
And so we talked about what she'd found. I told her the kinds of things we were doing in yeast, and it seemed like it would be very simple to do an experiment and see if the telomeres that she was working with would actually do the same kinds of jobs if we put them into yeast cells.
And so the question was, well, what if you put tetrahymena telomeres on this? Would this now allow the linear DNA to survive? And I thought that sounded like a cool idea, and I said, got to try it.
And so we just figured out how to do it and went ahead and did it. It was pretty straightforward, actually.